So this is a very quick tutorial on the macronutrients for not a nutrition class, but for a anatomy and physiology class. So I did not discuss a lot of steps in between. So this is just a very major information on the macronutrients. So carbohydrates. So there are monosaccharides, there are disaccharides, there are polysaccharides, there are starches, there are cellulose, there's a hemicellulose, so there's a lot of things. So let's look at carbohydrates as a polysaccharides, monosaccharides, and disaccharides. Not in order, but so we can also refer to carbohydrates as just simply sugars. So mono, right? Monosaccharides. So there are three monosaccharides. One is the glucose, another is fructose, and another is galactose. Glucose, fructose, and galactose. Monosaccharide. So we can refer to them as sugars. So what makes disaccharides? So disaccharides is just a combination of glucose, fructose, and galactose. So the famous disaccharide is a sucrose. Sucrose is just glucose connected through the glycosidic bond to a fructose. Glucose and a fructose. Another disaccharide is a maltose. So the maltose is just the two glucose units, glucose and glucose. So this is sucrose, this is maltose, and another is a lactose. It's just a glucose unit plus galactose, another sugar unit, right? Galactose. So in all of the disaccharides, as you see, you have a glucose unit. In sucrose 1, in maltose 2, in lactose also 1 glucose unit. That's what the disaccharides are. But when we look at the polysaccharides is just many glucose units, like 1, 2, 3, like this, right? It's a polysaccharide. More than 10 units, considered to be a polysaccharide. It's not an official structure, obviously. So, but that's what it is. Glucose, fructose, and galactose. So, disaccharides connected by the glycosidic bond, and we need a particular enzyme to break down this bond in order to absorb these units so we can use them for our system. We need for the sucrose, sucrase enzyme. For the maltose, we need maltase. And for the lactose, we need lactase. If you're missing a particular enzyme, that leads to a problem. Inflammatory reaction, allergies. So the people that are lactose intolerant, they either missing enzyme lactase or just too little of that enzyme that's produced by the body. So all of this are sugars, or saccharides, or carbohydrates. So then there's also our starches. Starch is a polysaccharide made out of the bunch of glucose molecules, made out of the bunch of monosaccharide. So let's say that each circle is a glucose molecule, right? Like these diamonds, right? Whatever. Glucose molecule. So the starch is a polysaccharide is the form of sugar that's stored in the plant products. That's a polysaccharide. Then there is also cellulose. Cellulose, also made of glucose molecules. Let's put fiber here. Part of the plant structures, also glucose molecules. Whatever, that's not very scientific what I'm doing right now, but it's basically glucose molecules. So that's the fiber polysaccharide, that's a starch polysaccharides, right? This is the storage form of sugar in the plants, and it's also part of the plants. This is the structural form. So when we're consuming plant products, we're able to break down these bonds between glucose molecules because we do have enzymes. But we're not able to break down these bonds between cellulose structures within fiber because we're missing on a particular enzyme that breaks down these bonds. We're able to break down maltose because we have an enzyme maltase. We're able to break down sucrose because we have an enzyme sucrase. We're able to break down lactose because we have an enzyme lactase. Those who are missing a particular enzyme cannot break down a particular bond. Bond is a lock, and enzyme is the key that needs to unlock that particular bond. Those people who are lactose intolerant, they're missing a particular enzyme called lactase. For starches, let's say we need amylases to break down amylopectin. I don't want to get there, but. And there's also another type of polysaccharide, which is a glycogen. Glycogen also a bunch of glucose molecules connected one to another, right? Again, it's not very scientific what I'm doing right now, but it's just a bunch of glucose molecules connected one to another, and that's what makes a polysaccharide. More than 10 sugar units is a polysaccharide. So the glycogen, it is the storage form of sugar in humans. We can store glycogen in the liver, right? And in the muscle, right? And we store them in the form of polysaccharides. The same thing happens in the muscles. Why do we store it here? So we can use it later on at night, for example, during physical activities, when you're not eating, when you're starving. So that would be a supply of sugar to the blood. So starch, we store it in the plants, but we can consume and digest this. And cellulose we're not able to digest just because we do not have a particular enzyme. And the glycogen is just a form of sugar that we store in the system. So basically, food that we consume, let's say we eat starch, right? And that's polysaccharide, right? Complex. We break it down in a monosaccharide. Starch made from glucose molecules, right? So we digest and we absorb glucose. So this glucose can get into the cell where we split the glucose and we produce ATP. We can use this glucose to replenish, to restore glycogen. So we're basically using these glucose molecules that we just got it from the food, from the starch, let's say. So we can store it in liver. Why? Because when you're exercise or at night, when you're starving, we need energy. Energy is not coming from food because you're not eating. So the energy in blood or blood glucose drops down. So we can break glycogen back into the glucose molecules. So we can bring this glycogen back in the circulation. If we consume too much of glucose, right, then we can convert it into fat and store it in adipose tissue. But when we will convert it into fat, it will become a triglyceride. That's next. Once it is a fat, it is fat. You cannot revert this back into a glucose. Also the sugar, right, we can store it in the muscle, also in the form of glycogen, right? Let's say like that. Again, not scientific structure. It's just a bunch of glucose molecules connected one to another. You see liver glycogen, we can break down through glycogenolysis and release those glucose into the blood. Muscle glycogens, we cannot. We will keep this glycogen, we will keep this glucose molecule specifically for muscles functionalities. Proteins or peptides. So proteins are made from amino acids. This bond is called a peptide bond. These are the peptides. You're making a protein. So the same analogy, the proteins or the peptides are the polymers, and the amino acids are the monomers. So the amino acid is a monopeptide. Bunch of amino acids together, you have a polypeptide. So let's say this is a polypeptide. You need an enzyme, let's say proteases. There's a numerous of proteases that have their own name to break down this polypeptide chain, to break down this bonds so we can absorb amino acids, single file. So to put in comparison with the carbohydrates, when we consume proteins, this polyform, we will break down this into a, let's say, single amino acids that will end up in the blood. Yes, we can use this for energy, but usually your body wants to use this for growth and structure. So let's say we want to rebuild muscle tissues or rebuild cells, right? So it gets into the cell as well. So the cell can use this as a structure and material and can build something within the cell. If it's too much, right, then this amino acids will end up in adipose tissue and would be converted into fat, into a triglycerides. However, the major functions of amino acids in the system is a structure or material for growth and structure. Lipids. Lipids can come in different shape and form. They could be long-chain fatty acids, short-chain fatty acids. So let's look at triglycerides. Triglyceride. This is a glycerol, a sugar that holds 3 fatty acids. Fatty acid 1, fatty acid 2, fatty acid 3. When we digest this through food, right, we bring this exogenous triglycerides into the system by removing fatty acids. So when we digest this, then in the blood, what's end up is free fatty acid 1, free fatty acid 2, free fatty acid 3. We can use this to bring it into the cell for energy. We can use this for energy, or we can use this as a material structure to rebuild cell, or to use this as energy. If it's too much, we bring this into adipose tissue and we will reform triglycerides. We will store it back as a triglycerides. However, fats comes in different shape and forms. So there are saturated fatty acids. If one carbon has all 4 bonds taken. So this essential carbon is saturated. That's why we call this saturated fatty acids. Let's say this carbon also saturated on all 4 sides. That's what the saturation means. Then we have unsaturated fatty acids. If there's a double bond in between carbons. If it's one double bond, we call it monounsaturated fatty acids, MUFA. If there are two or more double bonds, it could be two, could be three, we call it polyunsaturated fatty acids. As a rule of thumb, saturated fatty acids usually are solid at room temperature. Monounsaturated fatty acids usually are liquid at room temperature. And polyunsaturated fatty acids are definitely liquid at room temperature. So the temperature can change the state of a particular lipid. So this at the room temperature is solid. Think of the saturated fatty acids as a butter. At the room temperature is 72 to 74 degrees of Fahrenheit. Right? When you increase the temperature, the solid will become a liquid. With MUFAs and PUFAs, when you decrease the temperature, this liquid will become solid. For the PUFA to become solid, you really need to decrease the temperature, and most likely you have to go to commercial freezers when the temperature is approximately, let's say, minus 25 Fahrenheit to 40 Fahrenheit, and it will most likely still will be a liquid form. But if you will go down with the temperature, it will definitely solidify, because all of the lipids, they share the properties. So let's say if you take a seed oil, right, you will have a lot, like if this is a scale, you will have a lot this, or PUFA, let's say a little bit of MUFA, and a little bit of saturated fatty acids compositions. That's why at the regular temperatures it still will be liquid, but you can solidify at the low temperatures. They share the properties. Same thing with saturated fats, same thing with MUFAs, so it's just the properties changes. Double bonds are being considered weak bonds. Single bond is being considered a strong bond. The double bond is basically you have carbon, and you have hydrogens on one side. So there are no hydrogens here. So that's with the double bond, you'll have something like this, kink. It will bend, right? Because here hydrogens prevent bending. Positive, 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 positive. They repel one another. Here, nothing here. So they will bend. So when this chemical structure creates this bend, like that kink, that prevents from lipids stacking one on top of another. With saturated fats, they stack one on top of another because they're kind of linear. But it will become a liquid when you will increase temperature. So the double bonds are the weak bonds. It means that when you increase the temperature, it's liquid at the room temperature. When you increase the temperature, it's easier to break down. When you go above the smoke point in cooking, you can break down this double bond and it will become carcinogen. Same thing with PUFA. The more double bonds, the more breaking points. So that's weaker when you increase the temperature, but it's very strong when you decrease the temperature. And it is important in health, especially in cooking. So this is a very quick tutorial on the macronutrients for not a nutrition class, but for an anatomy and physiology class. So I did not discuss a lot of steps in between. So this is just a very major information on the macronutrients.